Hey guys, thank you for coming out this early in the morning. Um, if you guys could please turn on your webcams at some time, that'd be really great. I guess it's pretty early. We've already had one lecture, and I guess, you know, I know it's tough, like 3 lectures in one day, but it was like it was the best that we could do to accommodate all of the speakers and their schedules. After all, they are world leaders, and they have had a lot of meetings and everything. One of our speakers actually canceled his lab meeting in order so that he could speak with us. So thank you all for coming out and just being here. That's interesting. Brew, is that a picture of your ceiling? Lol. And Catherine, could you please turn on your webcam? Well, when you can, and then we can all see each other. I'm sure you guys can see me in my scrubs. Huh, now we have like a dancing ceiling with Brew's picture. Yay, thank you. Okay. Alrighty. Let's see. Oh, hey Catherine. How are you doing? It was great seeing you at what was it, Swirl Red Mango the other day? I can't hear you. I can't hear you at all. Can you hear me? Okay, yeah, I can't hear you at all. Okay, I just unmuted you. I don't know what's going on. Do you want to try calling in? Because, like, I can see you moving your lips, so I know you're talking. And I guess it could be your lips, but maybe, like, maybe Dr. McShane might not be able to as well, or something. Yeah, sorry about that. When you get a chance, Catherine, can you, like, try to, test the speaker?
Yeah. Like,
Oh, that's great. Okay. Okay, I
Okay.
can hear you. Awesome. Thanks. Brew, just a quick question. If you were to ask a question to Dr. McShane when the presentation is going on, please, please, if possible, like show your face or what is that? A pen, or like maybe maybe like give me the question or something, because then like we can put a face to a question. You know what I mean? Like rather than a machine or the computer asking, it's like actually a person, right? I mean, we don't want any robots here. I don't think Big Brother has come yet or whatever. Yay, Dr. McShane is on. Okay.
Hi, can you hear me?
Yes, Professor McShane, thank you very much for coming. I'm going to make you the presenter right now.
Okay, I've realised that I don't have a webcam on my desktop, and I haven't got my laptop on me. So, were you expecting me to do this as a video conference or as a teleconference?
Well, if you aren't able to access a webcam, that's okay. We'd just like to be able to hear your voice, and also that you can control your slides and switch to move to the next slide or something. Or if you're not able to move to the next slide, you can. I can be the presenter, and you can just tell me next slide or something, and I'll move.
All right. I'm sorry. It's been a rather complicated 24 hours, and I didn't think to bring my laptop in today. We could have been done as a video con. But if you're comfortable with me doing it as just speaking, then I think probably that's easier.
Okay. If possible, could you please try to see if you can try to change the slides or switch slides or something?
Sure.
There's an arrow, I don't know if you can see, but at the top bar, just below the tabs for quick start meeting and meeting info, you can actually switch the slide. It says, like, for example, right now we're on slide number 1, and you can click the arrows to change.
Tell me again where the arrow is.
So if you see on right below, there's a bunch of tabs that say quick start meeting info. And
oh yes, I'm with you. Sorry. Okay, so that one is. Oh, I see. I'm with you. Yep. No, that's fine. I can advance the slides. Yeah.
Okay, that's great.
Brilliant. Okay, so I'm just going to go and grab. I was just getting a glass of water, so just bear with me 2 minutes. I'll go and grab that now. I wanted to dial in.
Okay, sure.
Sure you can hear me. So you can hear me okay, can you?
That's perfect. Thank you. And I do not hear any echo or anything. Thank you.
Fantastic. Well, I suggest we leave it. I apologize for not being able to see me in person, but I think it's probably easier to leave it like this.
It's fine. Don't worry.
Okay, great. Thanks. I'll be back in 1 minute.
Mm-hmm.
Hi, so I actually do have a webcam now, this borrowed one.
Oh, that's great.
Would you like me to try? I'm sorry that you've got to this earlier. Would you like me to try and plug it in?
I'm sorry, I can't hear you very clearly.
Can you? Okay, so let me. I'm hoping webcam is going to work. Now maybe I need to change where I'm connected. Hi, can you hear me? So I've plugged the webcam in. Hang on.
Mhm.
Yes. Bear with me. Okay. Yeah. I'm just trying to install this webcam. It's almost done.
Mhm.
There we go. So that should. There we go. Can you see me and can you still hear me?
Yes, I can see you very well. However, I can't really hear you that clearly.
Move the webcam closer and then get. I mean, if it was better just to hear me, then I can go back to just the other option, whichever you think is better. Is that clearer?
Yeah, that's very good. Thank you.
But it probably doesn't. I mean, you probably don't want to be staring at my neck while I'm talking. So, is it? You tell me which you prefer.
It's clear. Yeah, I can hear you.
If I put the webcam on top of my computer so you can sort of see me, then can you still hear me, or is that not so clear?
Oh, I can hear you very clearly. Thank you.
Okay, so should we go with the webcam then?
Sure, that'd be great. Thank you.
Okay. I'm now just going to do one last thing, which is lock my office door so that to stop people thinking I'm in my office and they can come and talk to me. Okay. So, is that all right?
Yeah, that's great. Thank you.
Okay, so you tell me when you're happy for me to start.
Okay. Right now we have 1, 2, 3, 4, 5, 6. I think we should maybe wait for just a few more minutes so that a few more students can come, and then we'll.
Absolutely fine. And when, I mean, normally when I give lectures, I suggest that people interrupt as we go through and ask any questions as we go through, because I think that's more useful. I guess that works here because people could send me a message, or is it easier to say we do this question at the end? How do these normally work?
So basically, our system is that we will have a 1-hour talk with you, followed by a half an hour of question and answer time. And the way that the students do this is that they first send their questions to me, and I will introduce each student who wishes to ask a question and give them the time to speak. as of right now, I've muted all of the students so to minimize audio feedback.
Okay. Absolutely fine. All right, so I'll just give a straight lecture. So I've got quite a lot of slides for an hour, but it's fine. Yeah, no, that's fine.
Mhm. 1, 2, 3, 4, 5, 6. Okay.
Oh, that's 53, actually. That's not too bad.
Mhm.
Yeah, no, that's fine. Okay. So, well, you just tell me when you're ready for me to start.
Okay. Let me plug in my computer real quickly. And anyone have any questions? Remember, students, please type them into me. And during the Q&A session, I will make sure to basically just moderate and ask questions and answer questions based on timing and everything.
Okay, brilliant. Thank you. Sorry,
Mhm.
technically incompetent about this.
And I just want to say thank you for taking the time. Like your schedules must be really, really busy, and we'd just like to say thank you on behalf of IHN,
No, no,
the IHN team.
pleasure. You know, this is something I like to do.
Okay, Dr. McShane, Professor McShane, anytime you'd like to, please feel free to begin. Um, I will, if it's okay with you, I will first introduce you and talk a little bit about your background, and then we can get started. Would that be good?
Fantastic. Please do. Go ahead.
Okay, so today we have the pleasure of having a special speaker from actually the other, well, on the other side of the, perhaps the Greenwich timeline, or the Greenwich line maybe. I'm not exactly sure. I may have forgotten part of my, I guess, 9th grade geography. But today we have Professor Helen McShane, and she is a professor, the professor of vaccinology at the University of Oxford. There she leads the TB vaccine program. She trained at Guy's Hospital in London and was awarded a training fellowship in 1997. And at Oxford, she obtained her PhD investigating prime boost immunization strategies in mouse models of TB. And she was awarded a fellowship in 2001 to evaluate immunization strategies. And she's also a consultant in HIV and genital urinary medicine and received another fellowship in 2005. And she's now a full professor, and she's a fellow of the Royal College of Physicians in 2008 and was appointed that in 2008. So she is very decorated and famous, and we are very glad to have her here today to talk about global process in TB vaccination development from all of her journeys and experiences throughout, you know, her what she's seen all these years, and perhaps give us a lot more insight into what's going on exactly in basically TB right now. So students, I just want to say first, thank you for joining us, and Dr. McShane, please feel free to Come on in.
Fantastic. Thank you very much. Thank you for the invitation to talk. So really what I hope to do today is give you an overview of TB vaccine development, give you a bit of background to the field, give you a sense of why we are where we are, review what the major different strategies that are being pursued throughout the field is, and then towards the end I will focus on the work we've been doing in Oxford, really partly because it's what I know most about and partly because it illustrates, I think, very well the pathway for development of TB vaccine. So, and I understand that there's a question and answer session at the end of store up the top. Okay, so this map, which is taken from the WHO, really just illustrates where the global burden of TB is. And I think the first thing to say is it's everywhere. And you know, TB is not a disease that respects geographical boundaries. But you can see that overwhelmingly the burden of disease in terms of incidence is overwhelmingly in sub-Saharan and southern Africa. There's a lot of disease in India, China, Southeast Asia, former USSR as well, but rates of TB are going up. Okay, so epidemiology of TB in the 21st century. So the WHO figures from 2011 are that there were 8.7 million new cases in that year and 1.4 million deaths. So, you know, a staggering burden of both mortality and morbidity. The emergence of drug-resistant strains of TB, particularly multi-drug resistant, extensively drug-resistant, and we now have totally drug-resistant TB, has really compounded the problem, meant that our current control measures have become less and less effective and adequate. The WHO described XDR-TB as virtually, and it's salutary to note that in the big outbreak where we defined XDR-TB for the first time in 2005 in KwaZulu-Natal, 48. Clearly, the geographical overlap with the HIV epidemic has had a devastating impact. If you are infected with HIV, you are more likely to get TB, both to reactivate latent TB and to develop new TB infections. And we know that treatment with antiretroviral therapy does not bring us back to an HIV. And overall, we know that about a third of the world's population are latently infected with TB and are at risk of reactivation should they become immune. So this slide really just illustrates that this is a relatively old slide now, taken from 2007, showing every red dot is a country where XDR TB isolates have been identified. So this is extensively drug-resistant. And there are two things to take from this. You will hear me okay? Is that okay? There are two things to take from this. The first is that the red dots are everywhere, and XDR TB does not respect the boundaries. And the second is that the lack of red dots in particularly sub-Saharan Africa is of course not because there aren't XDR TB strains circulating. It's because those laboratories tend not to have the facilities to test them. So how on earth do we start tackling this epidemic? Well, in 2006, the WHO put out this document called the Global Plan to Stop TB, which sets some terribly ambitious targets taken from the development point of view. But what was important about this document from a vaccinology point of view was that for the first time there was explicit recognition by the WHO that we were not and would not control the current TB epidemic with the use of the currently available tools, largely the drugs that we have, and directly observe therapy, so watching people take. And that we need new tools to really get on top of it. We need new drugs, of course, because of the drug strains. We need new diagnostic tests so that we can diagnose people more quickly and reduce transmission. But ultimately, the only cost-effective way to ever control any infectious disease outbreak is with effective. And I think just a note at the bottom, it's salutary to note that whilst it's important to have ambitious targets, even at inception, this plan was less than funded, and that was, of course, before the disastrous current pandemic. So the next slide tells you a little bit about BCG. So BCG is the currently available vaccine against TB. It's the only licensed vaccine. and it is live attenuated Mycobacterium bovis, strain of Mycobacterium. It is the reason why we class BCG has been around for almost 100 years. It first developed in 1991 and was first given orally. And there have been many, many studies over the last 50 years in particular looking at the efficacy, looking at how good BCG is at protecting babies. We know that when given at birth, as BCG is throughout the world, BCG is pretty good, confers about 80% protection in protecting against severe disease, disseminated disease, and tuberculosis. And it also protects against leprosy. But what BCG doesn't do with any sense of consistency is protect against lung disease in adolescents. We also know from studies done by Laura Rodriguez at the London School, and this is an amazing paper, that if you boost with BCG, and you give BCG more than once, that doesn't improve your protection against TB. So we need something better. So this slide here, which is a cartoon of the various different efficacy trials that have been, really gives you a sense of the variability. So in the top panel we have control trials, which are methodologically more rigorous, and in the bottom panel we have observational studies, less methodologically good trials. And you can see here this is 0. So any trial in which confidence interval span 0 is going to be didn't work. And you can see that there are some trials, the British MLC study here, which was probably the best trial ever, showed it to be very effective. But there was a huge study in Shinglepuss in South India showing BCG really did nothing. If anything, had slightly negative. If you do a meta-analysis and you combine all these studies, then the average efficacy is about 50%. But really that's pretty meaningless, and the efficacy spans 0 to 80%. And we know from these studies that latitude has a major influence. So we know that the closer you live to the equator, less likely BCG is to work. Perhaps that gives us a clue as to the mechanisms by which BCG doesn't work. So why doesn't BCG work is a really, or why is it not consistent, particularly in TB endemic countries, is a really important question. Because we hope that a new TB vaccine will overcome the limitations. We know that there are many different strains of BCG that are used throughout the world, and we know that there are sequence differences in those strains of BCG. What is much less clear is whether those sequence differences are responsible for varying any differences. There are some animal models to suggest that they do, and some animal studies to suggest that they don't. But we certainly know that the same strain of BCG has been used in different trials and works in some and not others. That's what we only know. We know that nutrition is important, no vaccine and but many of the areas of the world where BCG or TB is most endemic are not necessarily the most malnourished. That alone is not enough. And really the explanation that has gained most evidence over the last 5 to 10 years is that exposure to environmental mycobacteria, so pathogens from the same family as TB, that don't cause disease unless your family is expressed, either mask or block the effect. So what do I mean by that? Well, by masking, what I mean here is that we know that BCG induces some, we know that exposure to environmental mycobacteria induces some immunity, and essentially BCG doesn't seem to be able to increase that level of immunity. So here, this is data presented from work at Hazel Dockwell's group at the London School, where she took adolescent children who hadn't had BCG in Malawi, in Africa, and in the UK. And she looked at the baseline immune responses, she then gave them BCG, she then looked at their immune responses after BCG. And what she found was in the UK, children had very low baseline responses and very high responses after that. In comparison, in Malawi, the children had very mixed and moderate responses before vaccination, presumably induced by environmental. And BCG didn't boost those responses. So you can't enhance these responses by giving BCG. The other hypothesis is a more active one, is what we call blocking. So the idea here is that BCG is a live replicating that. It has to replicate in order to work. So if you do BCG, it doesn't work as well. And if you have exposure to environmental bacteria, which gives you some pre-existing immunity, that immunity can inhibit the replication of BCG and therefore stop BCG and therefore stop BCG. And it's likely that these two explanations both work together. So there are other problems with BCG. We know that BCG, as I've just said, is a live attenuated vaccine, and therefore it's not safe in people who are immunized. Always been contraindicated in adults, but up until relatively recently, it used to be recommended to give it to BCG vaccinated babies, or to give it to babies, on the basis that the balance was it was better to protect them against TB. We now know from work in South Africa that there is a very high risk of those babies developing a disseminated disease, the BCG that's spread out. And for that reason, the WHO have changed their policy. It said that BCG should not be given until an infant HIV status is in areas of the world. I mean, that's fine to decide in Geneva, but the reality on the ground in Africa is that it's very difficult to do that. You often don't get infants coming back to have their HIV negative status confirmed, and therefore they often miss their BCG completely. And in most areas of the world in Africa where I collaborate with, BCG is still routinely given, even to HIV patients, because of the relative risk balance is that it's still better. It sort of illustrates difficult issues. So, okay, the other thing we need to think about in trying to develop a new TB vaccine is understanding a bit about the vaccine. So we know that of 100% of people who are exposed to TB, about 70% of them will actually clear TB very quickly with a very vigorous, innate immune response. And really, you know, if we could mimic that with a vaccine, that would be ideal. But we're not at a point where we know well how to use innate immunity. Of the third of percent of people, or 30% of people, or third, who are infected with TB once exposed, about 90% of them go on to control their latent TB very nicely and only reactivate in about 5 to 10% over a lifetime, providing them. And again, if we could understand what those latently infected people were doing to control that TB, then perhaps that's what we should finish with. So what aspects of immunity do we know are important? So we know that class 2 restricted CD4 T cells are essential. We know that from the animal models, but of course we know it best from HIV infection. As your CD4 your risk of death goes up. We know that cytokines such as interferon gamma and alpha are essential. We know that again from mouse studies, but we also know it from human studies, from people with genetic defects in the pathways, the people who are starting on infliximab, a drug used to treat rheumatoid arthritis, where you block TNF. And we know that that can then lead to a reactivation of latent. We know there are other aspects of immunity that's important for positive CD8 positive T cells, and the delta T cells, CD1 restricted T cells, and newer cytokines. But really, I think the challenge is developing modern day vaccine technology. There's not a huge role, at least in the current thinking, for B cells in antibodies, although I think most people would say that they do have a part to play, not as important as cellular. So putting all that together, how do we design a new TB vaccine? Well, first, because we know that BCG does confer protection, first 10 years of life, really everyone in the field is incorporating BCG or in some shape or form. BCG is being included because we want to hang on to what it does do rather well. We need a vaccine that induces primarily a strong cell-mediated immunity. And broadly speaking, there are 3 ways to do this. You either leave BCG as it is, given at birth, and then develop a subunit vaccine to be given at a later time, which could either be a protein and an adjuvant or could be a recombinant virus. Or some groups are replacing BCG with an attenuated or an improved strain of BCG. So they're suggesting that actually replacing the prime is important. Ignoring those noises. And clearly you can put the two together so you can enhance the responses. So this pipeline just illustrates where we are at the moment with TB vaccine. It's a little bit out of date. This was last year. It's taken from the TB vaccine blueprint that was published last year and really illustrates the lead candidates in vaccine. So you can see that there's a vaccine called MW, about which not a great deal is known, but is being tested in studies. You can see there are two candidates in phase 2B. That means efficacy. Actually, does it work? One is the vaccine developed by Oxford, MVA, and one is vaccine developed by Helen, which is a adenovirus. And then there are vaccines in phase 2, phase 2 immunogenicity testing, and some earlier candidates. So there are about just over a dozen candidates currently being tested. Okay, so to talk through that just in a little more detail, and the details here I think are not important, it's really the principles. So thinking about recombinant BCG strains, so these are strains of BCG that are being developed to replace BCG. So RBCG30 is a recombinant BCG that expresses an antigen B and has been designed to replace this. First went into clinical trials in 2004 and is not currently being developed for various reasons. This vaccine is a bit more interesting. It's known as VPN-1102. This is a vaccine that's being developed by Stephen Kaufman's group and is a recombinant strain of BCG which expresses listeria lysin. The idea here is that the listeria lysin increases the acidification of phagosome, increases endothermal escape of antigen, and therefore increases access to the one presentation. So you get a better CD8 T-cell response, at least that's the hope. With this vaccine. There are phase 1 studies in Europe and phase 2A studies in babies that are currently ongoing. And then RS422 is an example of putting these two strategies together. So this vaccine expresses lysin, so similar molecules to lysin, but also expresses some antigen. That vaccine went into clinical trials about 18 months ago now and has now been withdrawn because 2 of the subjects who got the high dose developed shingles. We don't know if that was related or not, but vaccine.
Dr. McShane, sorry for interrupting, but several of the students are having trouble hearing you. I was wondering, could you try to perhaps speak closer to the mic, or perhaps even call in from a phone or something?
Okay, so if I speak like that, is that clearer? Or I can unplug the webcam and use the microphone without the video as we did before.
I think it's as of right now, this is good. If possible, if you would like, you can just turn off the webcam, and we can turn it on later or during Q&A if that's good.
It's no trouble. I mean, I can angle it so it's through the window. But can you hear me okay now?
I can hear you clearly now. How can we try to continue the presentation this way, and then if students are having troubles, please send me a message and I'll see what I can do about it. Thank you. Okay, thank you.
So, okay, moving on to attenuated strains of TB. So this is another example of how to replace BCG. And really the idea here is that BCG, which is attenuated microbiome bovis, is the wrong starting organism, and we should be starting with TB. So there are 3 groups looking at this, or well, in fact 2 groups. So Martin and Zaragoza has the P mutant, which has just gone into clinical trials in Lausanne in Switzerland. And then Bill Jacobs at Howard Hughes has a whole library of oxytocin developed some of which he would like to put forward into trials. And this one, I plus as an example of one of these. This is in fact a manufacturer and artist expressing the EFX secretion from ED. The important thing about I plus, which was published about 18 months ago now, is that this vaccine is the only vaccine that has ever induced sterilizing immunity. It doesn't do it all the time, but it does show us what is possible. Okay, so now I'm going to give you an example of some of the booster vaccines. And these are vaccines not designed to replace BCG, but designed to be given after BCG. So the first is developed by GSK. This is called M72 now, and it's a fusion protein with two antigens, the 32 and the 33 antigen, with the AS01 adjuvant. This is the same adjuvant that GSK is using for malaria. This vaccine went into clinic in 2004 and has since been in a number of studies. And we know from all of those studies that this vaccine induces moderate levels of antigen-specific CD4 T cells. So there's currently a phase 2B efficacy trial in adults, which is being planned. Can I just check? Can you hear me okay now? Is that okay? Yeah. Okay. Study yes. Okay. So fusion proteins, so the other main group looking at fusion proteins as a way to boost immunity is SSI in Copenhagen. I have three different units. So the first is ESAT6 and 75B. And the problem with this is that, and I don't know if you've had any talks on TV diagnostics yet, ESAT6 is one of the antigens that is used in the new diagnostic test, IGRAs or inflammatory pathways. T-spot TB and quantitative goals. The problem is if you put those antigens in a vaccine, then you're going to confound your dose. And even SSI recognizes now, so there's a place to be with TB24 in the next generation. And then finally, there's a hybrid 56, which is a third generation vaccine, which is designed to be a latency vaccine given to patients. Okay, and then the other way to boost BCG is to use viral vectors, so which are a very good way to deliver antigens. And there are two different viral vectors being looked at the moment in this field. So the first is what we call ERAS-402. This is a recombinant adenovirus. It's AB and T4. And what's interesting about this vaccine is it induces high levels of CD8. And there's currently a phase 2A study in infants. And then the other vaccine that is a viral vector that's in the field, and these two last vaccines are really the lead candidates in the field, is the vaccine we developed in Oxford, MVA-85A. And this is a recombinant modified vaccine and another viral vector. So we're using the virus here, delivery system, to deliver the virus and expressing antigen. And again, this vaccine has been designed. So what I'm going to do for the second part of the talk is talk through MVA-85A, the data we have, the results we have, really as a way to illustrate the pathway for the development of these. So there are 4 animal models of disease: mice, guinea pigs, non-human primates, and cattle. And we have demonstrated in all 4 of those animals that you can improve BCG-induced protection. So here in mice, here you can see the survival in guinea pigs. So, excuse me. Here you can see some representative lungs from animals vaccinated either with saline, with BCG, or with BCG-MVA. And here you can see in cattle, there are increased numbers of disease-free animals in the BCG-MVA group compared to BCG alone. Can I just check that I can see lots of activities? Can I just check that you can hear me okay? I don't know the name of the coordinator. Can you hear me? It's all right now. Okay. Okay. So this slide shows some Ellie spot data. So the advantage of the animal models is, as well as looking for protection, we can use these animal models to try and identify immune carriers, which then allow us to determine what kind of immune response we can give. So here we've seen that animals that were protected in this couple of experiments have higher levels of interferon gamma and their Ellie spot responses than animals who are not protected. And that's important because gamma is one of the main readouts. We also, in the same experiment, found that levels of IL-17, as well as levels of were important. And again, that's important because those are parameters of look at. So moving on to the clinical program, this slide summarizes the clinical trials that we've conducted with this vaccine. And really just to illustrate how you go about testing an ETB vaccine, we start with volunteers, we then look at latently infected people, we then moved into HIV infected people. We started in the UK, and when we had safety data in the UK, we then moved into here we've been studied in Gambia, in South Africa, and most recently in Senegal. And all these phase 1, phase 2A, safety and immunogenicity studies lead into these efficacy to look at actually does the vaccine actually work? Do we actually stop people getting? So the outcome measures in all of these trials, primarily safety. We also look at immunogenicity using a variety of different parameters. And ultimately, the bottom line is we look at efficacy. The vaccine actually stop people from doing. What we see in all of those early studies is that this vaccine is highly immunogenic. So we see it here. You can see the kinetics of the Ellie spot response over time in BCG vaccinated volunteers. We see here that we induce highly polyfunctional responses. So T cells with lots of different cytokines, which we believe are important for function. Here you can see that in latently infected people, we see just the same kind of kinetics as in vaccinated people. And here, importantly, in HIV infected, we also see. So we then move the focus to South Africa, where we've had the pleasure of collaborating with the University of Cape Town for the last years. And we work at their clinical trial site in Worcester, kilometers outside of Cape Town. And what we see in those studies in South Africa is that this vaccine is also highly immunogenic. Here are the adult responses for the vaccine-induced responses, which are durable after a year after vaccination. Here are the adolescent children, and here are the infants. One of the things that's important when you consider infant vaccination is, can you give this vaccine at the same time? Because if you can, you're going to increase your uptake. So we did a study in Gambian babies looking at, can you give this vaccine at the same time? 18 childhood vaccines for babies. We had 3 groups of babies. The first got just their routine EPI vaccine. EPI and MVA together. Well, the third group got MVA alone, and the third EPI. What we saw was that the safety profile was excellent. We saw no effect of MVA on the EPI immunity, but we did see that when you co-administered EPI and MVA together, you reduced the MVA immunity. So there's the data. So you can see that the screening is no different, but at 1 week, 4 weeks, and 20 weeks after vaccination, you see significantly lower responses in the group that got MVA at the same time as the EPI vaccines compared with the group that got MVA. This is likely to be the alum in the EPI vaccines, reducing toxicity. And this data led to us giving MVA outside of the EPI schedule in our efficacy. So that's all very important. So the data to date shows the vaccine is safe, and the data to date shows that the vaccine is immunogenic, and we think it stimulates what we think is immunity. The bottom line is, does it work? Does it stop people getting sick? And we know, and this is work from the South African Vaccine Initiative, that the classical Th1 responses that we measure and that we know are important don't necessarily correlate with the. There are three main target populations for UTB vaccines: infants or babies in the developing world, adolescents or adults where the burden of transmission is, and HIV-infected adults who clearly are an important part. So just a slide on why TB vaccine efficacy is so difficult. So phase 1 and phase 2A studies looking at safety and immunogenicity, you can do in tens or almost hundreds. But in efficacy trials, we have to look for thousands of subjects and follow them up for 10 years. Because even though there's a huge burden of TB throughout the world, the disease incidence, even in high burden, is actually relatively low. And what you need in efficacy trials is a new safety. We don't have an immune correlate, so we don't know whether the immune responses we're seeing are the right ones or not. And we don't know which of any of the animal models fit. So that's why what we're left with is doing these kind of efficacy. So this is the design of our phase 2B efficacy trial that we've just finished and reported earlier this year. This was in South Africa babies. We had almost 3,000 babies. They were all BCG vaccines first. They were randomized to receive either MVA or placebo within a 4 to 6 month window after. This trial was powered to show 60% overview. So that's the paper. We published this in February, the results of this efficacy trial. And really, this was the first efficacy trial of a new TB vaccine that had been conducted. BCG was last tested in about 19. So this consult diagram shows the number of babies that we had in this trial. So we had 4,700. Importantly, already 281 of those, that's almost 6% of babies, 12 weeks of age, were excluded from this trial because they were already included, which really gives you a sense of the burden. We ended up with 2,797 subjects that were randomized either MVA or. So looking at the immune responses induced in this trial, so you can see here that we have in a small cohort, we looked at real-time immunogenicity. You can see that in the placebo group, there's no change. And in the vaccine group, we saw the induction of antibodies, as you would expect, and consistent with the previous. Okay, but the bottom line of this study was efficacy. And here was our data. So we had an endpoint one which was very stringent, which was infants either had to have culture-positive disease or they had to have TB diagnosed using a gene expert molecular-based test, or they had to have a very stringent quantity. We had 39 cases in the placebo arm, and we had 32 cases in the. That gives you a non-significant vaccine efficacy. With a confidence in 0. Same story was true of endpoint 2, which was slightly less stringent, and endpoint 3, which was an exploratory, which was any infant. We also looked at rates of quantiferon conversion, i.e. whether there was any evidence this vaccine prevented babies getting becoming infected with TB in the course of the trial. And again, we saw no evidence of any effect. 178 in the BCG MVA group compared with 101 for almost in the placebo group. So we didn't see any efficacy at any time. But one of the things that's interesting when you look at the length of follow-up is that if you look at all the follow-up, and the median here was 24 weeks, we had a vaccine efficacy of 70. If you truncate that follow-up at 4 months, your vaccine efficacy goes up a bit. And if you truncate it at 12 months from the post-hoc analysis, your vaccine efficacy goes up again. Now, the point of this is not to suggest there might have been efficacy there. We don't know. The problem is that it's not possible to detect early efficacy with the current design of the efficacy. So at 12 months, we only had 16 cases in the arm. And the reason why this is important is for these difficult targets, TB, HIV, and malaria, it may take us a few goes to get a vaccine that works. And if we can find a vaccine that works in a year, that isn't a deployable vaccine, but it helps us identify, helps us give us a steer that we're going in the right. So my view on this is that we should be thinking about redesigning these trials and recruiting much bigger numbers of people, but following them up for much more time, so that we can actually look at early efficacy to really give us that how we going in. Okay, so how do we think about these results? Well, I think we've described in the paper the immunogenicity. To be fair, it was comparable with the phase 2A data from infants. And it may reflect an immature immune system, but we know that the immune responses were about 10 times lower than we see in adults, and that may be important. I think what we now know that we didn't know before is that this level of immunity is not sufficient to improve protection of BCG in infants. And the really unanswered question is, if we saw 10 times as much as we do in adults, would that be sufficient, or do we need something? So, I mean, that's a sort of whistle-stop tour of where we are. To date, we now have 14 vaccines that have been evaluated in trials. We have 2 that are being evaluated in efficacy. And to date, we've identified no immunopathology in any of these. But I think the challenges going forward are vast. We still don't have a correlate, so we don't really know which vaccine is going to work or not until we do those efficacy trials. We don't know what the animal models mean, and of course this is a bit chicken and egg. We won't identify correlates, and we won't validate the animal models until we have there are real issues with the logistics of the endpoints, particularly in infants. And as I'm sure you can imagine, the world has a very finite view and So just in the last few slides, I want to illustrate where the direction my group is going and just thinking about ways we can improve the models. Because I think what we really need are better predictive models with which to select which vaccine to go forward. We can't put all of that kind of practice. So one of the first is using this thing called a mycobacterial growth inhibition assay. So this is an in vitro assay where you take blood or PBMC from someone who's been vaccinated, and you culture that blood with mycobacteria in a test tube in a lab. And you look at the ability of that blood or those blood cells to kill mycobacteria. These assays are used routinely for diagnosing TB, and they're used a lot in the drug field for drug development. And Bob Wallace, who's published a lot on this, has published showing that in BCG vaccines that some people do get a reduction in colonies, so a reduction in mycobacterial burden, a greater killing after BCG. Some people don't, and some people seem to after. But I think we've been funded by ARAF and by other people to look over the last couple of years at these kind of assays to try and see if we can use them as useful ways of measuring vaccine efficacy in a test tube before we have to. The other really holy grail to me is the idea of a human challenge. So we know that in malaria vaccine development, vaccine development, and influenza, human challenge models where you actually vaccinate people and then challenge them with the pathogen are used extensively to allow some demonstration of that before you use it. And obviously these trials can be then validated against it. And they can be used to identify. There's a problem with TB though, which is we can give people malaria, we can give people flu. I don't believe it is ethical to give people TB. But we can give people BCG. And BCG is a live replicating bacterium, which is licensed to the EU. And the idea here is that a vaccine that works against BCG might also work against TB. And the intradermal BCG challenge provided good model for what we'd really like to do but can't, which is aerosol TB challenge. So some work from one of my PhD students, Helen and tried to do this. So we gave people BCG, we took a punch biopsy from their arm. We looked at the amount of BCG we could extract out of And the idea here is then you might, once you set up your model, you can then vaccinate or not, and then challenge the BCG, and then look to see if you get a reduction in colonies in your group that have had your. And here you can see she did just that. So you can see that in people who had this before, there was less bacteria than in people who hadn't had it before, at least when we used PCR for this. Didn't see that when we looked at culture. This was a pilot study. There were lots of issues with the study, and I think we're now repeating this to see if this is reproducible and which of these two is. There are some issues about validation of this. So one important question is, does intradermal BCG represent an aerosol TB challenge? And we don't know. And of course, we can't validate that in humans because it's not give people an aerosol TB challenge. But we can give animals a TB challenge. So we can do that validation. We're doing that work at the moment. But the ultimate validation of this clinical model will be in a human. So I think my last slide is really just to show you what I think is the key for vaccine development. So we start with in vitro studies, we move into studies in small animals, mice, guinea pigs, we then move into larger animals, we move into apples, we move into the small scale, safety and immunogenicity studies, and then we move to those large, big efficacy. That's pretty obvious, but I think what's really important is that we take the data that we generate from these trials, feed it back to cross-validate into the animal model, into the human, so that we can actually iteratively work together to build better models to then select vaccines. Once again, because I think these are tough to pick, and I think we have to draw these out. We're really concerned with these. Good. Well, I'm sorry the sound didn't work to start with. I hope that was clear, and I hope most people could hear most of that. But I'm very happy to discuss it.
Thank you very much, Dr. McShane, for a very insightful and interesting lecture. I have a first request now for the Q&A. I'm going to be moderating this Q&A, and I will be asking all of the questions that the students asked during the presentation in the order from which I received them. And also, Dr. McShane, if it's all right with you, could we try to just turn off the webcam and just use the mic? Because I've noticed, like, at the beginning of your presentation, when you tried just using the microphone, it was very clear. And we can, if we need to refer to the slides, we can just refer to, you can just feel free to go to any slide if that's okay.
Okay, so what I'll do is unplug the webcam, and then I think it will click back into the mic. But yeah, I'm at the edges of my technical understanding of this.
Okay, thank you very much. Can we test? Okay, I guess that's very good.
So can you hear me now?
Yes, very clearly. Thank you.
Okay.
And I believe all of the students should be able to hear you more clearly now as well. Students, please tell me, chat, or send me a message if you cannot hear me, hear Dr. McShane clearly, or me. Yep. Okay. So we have quite a few questions lined up. So I will just be moderating all of the sound, and I will ask all the questions for the students. And some of the students, for example, it's still 7 a.m. or something, and
yeah,
so
no,
it's kind of crazy.
I'm impressed with the keenness, and I'm sorry I couldn't do this any later.
Oh no, it's fine. Like we totally understand. I mean, we're spanning maybe 8 time zones or something, and it's kind of crazy. So, okay. So the first question that came in was, with regard to the Andrew Wakefield controversy and the anti-vaccination movement, how, and you mentioned that there were different adjuvants used for vaccines and improvements of them. So I believe for students who don't know what an adjuvant is, just a little bit of background, it's it's a substance that's actually introduced into the vaccine and is given at the same time with the vaccine, and it's a medium that helps stimulate the body to be able to produce a more active response to the vaccine. And Dr. McShane, please correct me if I'm making any mistakes. But
Perfect. No,
I believe, okay, with regard to Andrew Wakefield, or former Dr. Andrew Wakefield, and the anti-vaccination movement, there was this whole controversy with the MMR vaccine and the fact that there was perhaps a correlation between a certain adjuvant, I'm not exactly sure what it was called, like temerazine or something, and autism. And since then, it's largely been disproven with subsequent studies and everything. And in fact, Dr. Wakefield's license was medical license was revoked because of it.
Indeed.
So I was just wondering, what this question, the student was saying that, what is your opinion of this, and with regards to the new adjuvants that are being introduced to TB, the TB vaccine?
Okay, so that's a good question, and there are lots of different answers to it. So in brief, I think it's really important for everyone to understand that Andrew Wakefield's research has been 100% utterly discredited. Many of the authors of that paper have retracted it. He, as you say, has had his license struck off, and I don't know anyone who is a doctor, a pediatrician, or a scientist who believes there is any issue at all. So that was really around MMR, which is a vaccine that doesn't have an adjuvant. It's attenuated viruses, and I think it's terribly important that the next generation of scientists understand that there is no evidence that MMR ever causes autism. Now I can talk for hours on why that is, but that is the bottom line. There's a different issue. So there have always been concerns about vaccine safety. There is a different issue, which was the thimerosal, or which is used as a stabilizer in some adjuvants. There were concerns as to whether that was safe. And again, there's really no evidence that I know of to support claims that it isn't. Vaccines are, what I feel is that these concerns are the luxury of the West. When I go to Africa and people understand that without vaccines you die and you get horrible diseases, no one says to me, gosh, they're concerned about getting autism. And I think, what happens is as diseases become less prevalent and people become less exposed to them, we become complacent and then we focus on real or imaginary concerns and things. In terms of TB vaccine development, there are, the BCG replacements don't have adjuvants because they're attenuated organisms and they don't need adjuvants. The viral vectors don't have adjuvants because, again, it's the virus that is, if you like, the intrinsic adjuvant. So it's only the proteins, and this was GSK and SSI on my slides, that actually have adjuvants. The adjuvants that are being tested are new adjuvants, and they're new adjuvants because we need them to stimulate T cells or cellular immunity, which is a different kind of immune response to the immune responses that the licensed adjuvants use. But to date, we know that these adjuvants are well tolerated. Is that sufficient answer?
I believe that's a very good answer. Thank you very much. Let's see. All right, the next question we had. And so the next question was, with regards to the trials you conducted on TB vaccines on children, why were children chosen as a group rather than adults? Because this question states that it seems to be safer to do these studies on adults and children rather than infants, because after all, infants, they are not able to produce their own antibodies until after a certain age, and they are more susceptible to a lot of infections as well.
Okay, so the first important thing is we don't think antibodies are very important in protection against TB. It's T cells or the cellular arm of the immune response that's predominantly important. And we know that infants can generate immune responses. BCG is given at birth, and we know that BCG works when given at birth to protect against severe disease. Infants were selected in this trial because infants are an important global burden of TB. Infant TB is a big problem, particularly in Africa. I think the field has swung since we've done this trial in thinking that adults and adolescents may be a more important target population in terms of transmission, and therefore that's why the focus of the next efficacy trial, which will be the GSK trial, is going to be in adolescents and in adults. So I think, that's an example of how our thinking has shifted a bit. And of course, our trial suggests that it may be rather difficult to protect infants. And that may be in part because of immature immunity. But infants, nevertheless, infants remain an important target population for a new TB vaccine because lots of infants get TB. And that's why infants were selected in this first trial.
Thank you very much. Okay, the next question is with regard to slide number 34. This comes partially from the last question. Why isn't the elderly population included, basically, in the trials instead of the children and other groups? Like, especially because the elderly population is considered rather one of the more immunocompromised groups, since after all, perhaps a lot of the vaccinations they've received as children and infants have largely had their impact and immunity, capabilities decreased. For example, is a possible explanation because there is some sort of inherent immunity due to their age or perhaps more encounters with other similar diseases that have given elderly people some sort of immunity?
So I think the simple answer is that although, yes, of course, as people's immunity breaks down as they get older, they can reactivate TB. In terms of a target population, those numbers are pretty small. So when you look at the epidemiology of TB, there's a peak in infancy, which is why we were interested in infants. And then in childhood, rates of TB globally go down quite a lot. And then the big burden of disease is in adolescents and young adults. That's where the numbers are. So really our development program and the trials that we do are directed towards those target populations. So the three target populations, infants, young adults, and HIV-infected people, globally represent the biggest burden of TB. Those are the groups that get most of the TB, and therefore the trials are geared towards testing the vaccine in those populations. It doesn't mean that if once we have a vaccine that works, it would then be deployed in other age groups, other risk groups, which are at lesser risk, but nevertheless still at risk.
All right, thank you very much. Let's see. The next question states, how well does herd immunity work with regard to the BCG vaccine? Just as a little bit of background information for students, herd immunity is basically where individuals who cannot be vaccinated, especially with live attenuated vaccines due to their immunocompromised state or allergic reactions to antibiotics such as neomycin, must rely on the fact that other people in the community are already vaccinated and will not give the disease to those unvaccinated people. Like stated again, the question is, how well does this herd immunity work in regards to the TB vaccine?
Okay, so, I mean, to a certain extent, so herd immunity requires that we have an effective vaccine, and we don't have a very effective vaccine. So to a certain extent, it's a null question in that we don't have a vaccine that even works to the person we give it to, let alone, and how if it doesn't work reliably to the person we give it to, then it isn't going to do anything in terms of herd immunity. I mean, that's a rather simplistic answer, but so there are populations. So in the British MLC study in the UK, BCG works very well. And if in populations in which it works well, and it works well particularly in temperate climates, where of course there isn't much TB anymore, then it will reduce rates of TB in adolescents and adults. Therefore, it will reduce transmission rates, and therefore there will be an effect on herd immunity. But it's only, there are very few settings where we see such good efficacy against pulmonary disease, and it's really only in the British MLC study and the North Alaskan Indian studies that we've seen those. In any other population, particularly in Africa, in India, and China, BCG doesn't seem to protect against adult pulmonary disease, and therefore would stop people developing severe positive TB and therefore transmitting. So there will be no effect on herd immunity.
Thank you very much. All right, the next question is in regard to slide 38 with the flow of patients during drug trials. So the question is, how do you determine which patient's information to keep and which to remove from the actual final data analysis? Because in all, about 125 people couldn't be followed up, 60 people withdrew consent, but only 3 people were not analyzed in the end. And these were all of whom who had incidentally received the placebo.
Okay, so I think the question is so there are 2 ways to analyze data. You do a per protocol analysis, or you do an intention to treat analysis. What we've done here is an intention to treat analysis. Sorry, what we've done here is a per protocol analysis, and we've excluded 3 subjects. So we excluded this person who got a dosing deviation, so we got the wrong dose, and there were 2 subjects here who didn't get the correct allocation or the correct intervention. So we excluded those from the analysis. Because there were so few subjects that we actually excluded, what that means is your per protocol analysis and your intention to treat analysis is essentially the same. In terms of these discontinuations and withdrew consent, we included, because you're always at the edges of power in a study like this, so we included all the data we could. So in the early discontinuation or the loss of follow-up, we included people until the point at which we lost them or they discontinued follow-up, because that data was important and relevant, even if that meant we lost them 6 months before the end of the study or something. And I think that's considered standard practice.
All right, thank you very much.
Okay. Just one more point on that.
Sure.
I think there is a difference here. This is a vaccine trial where they got a vaccine at time 0 and were then followed. I think, therefore, what I've just explained and justified is fine. I think it is different if you've got a drug trial where you've got different rates of discontinuation, where clearly then there's a concern that there are different rates of discontinuation because of your drug. But that doesn't apply to a one-off intervention like a vaccine trial. And the other point was, you can see that the discontinuation rates, the withdrawal consent rates, were utterly comparable across the 2 groups. So that was the other thing that made us confident.
All righty, thank you very much. All right, the next question, slide 16. So in your opinion, out of the 3 recombinant BCG strains shown on that slide, which one, in your opinion, shows the most progress, or promise, rather?
Okay, well, it's an easy answer, really. This RBCG30 is no longer in development because there are regulatory issues with the way the construct was made. RS422 has been withdrawn because there were safety issues, so there's only one of these 3 that's in clinical development. I think there is some preclinical data that's been published on this vaccine, the animal studies, and there isn't yet, I don't think, any immunogenicity published from the clinical studies. To date, the safety profile seems acceptable, but I think, as we've seen with our studies, the bottom line is, does this vaccine protect against TB? I think there are real issues with how you do an efficacy trial with a BCG replacement, because essentially you have to do a non-inferiority study, and that needs a very big study, because essentially you have to give half your group standard BCG and half your group new BCG and compare rates of TB between them. And that kind of study needs big numbers. And I don't think we've quite worked out exactly how we'll do that in the field.
Thank you very much. All right. The next question is, why is it much harder to develop TB vaccines as opposed to other vaccines such as whooping cough, pertussis, or chickenpox, or varicella? For example, why don't we already have an effective TB vaccine?
Okay, so this answer goes for TB, but it also goes for malaria and HIV. So whooping cough, measles, pneumococcus, meningococcus, these are vaccines that are relatively easy to develop because we know what kind of immune response you need to be protected. And we know that that kind of immune response is an antibody response. So therefore, we know that we can test the vaccine. And if there are now vaccines, so pneumococcal vaccines and meningococcal vaccines, where these vaccines are licensed not on efficacy because you don't need to do efficacy. You can just show that they induce a certain level of antibodies. In contrast in TB, but also in HIV and also in malaria, we don't even know which aspect of immunity is important for protection, let alone what kind of level. We know that cell-mediated immunity, T cells, are important for protection, but we don't know what level or what particular type are necessary. We also, as I said, don't know what the animal models mean, and again, this is a bit chicken and egg. We won't know until we get a better vaccine. So that's why really what we're left with is having to do these very big efficacy trials, and to a certain extent having to make a best guess as to which vaccine should go into the efficacy trials.
Thank you very much. Okay, the next question is with regard to slide 5. Slide 5 shows a bunch of targets that were established for goals of these trials and hopefully the progression of the vaccine. And the question is, how initially were these targets actually established? For example, were they based on past results or what seemed to be the most feasible at the current time when these these targets were made, or was it something else?
That's a good question. I don't know where, so these targets were taken directly from the Millennium Development Goals. And I don't remember the background of how those were determined. I mean, it was a big global health forum. WHO were obviously involved. People like Gates would have been involved. The Global Fund, large multinational PDPs like that. But they were lifted directly from the Millennium Development Goals.
Okay, thank you. Okay, so the next question is, what is your opinion of different countries' different opinions on the BCG vaccine? For example, in the United States, currently the TB vaccine is actually not given. And so, for example, the student came from Asia where she received the vaccine as a child, and as a result, she had to go on a regimen of, I believe, rifampin and isoniazid for 6 months. So that, and in the end, she had to receive a chest X-ray to basically clear her. So technically, since for students' background information, technically, if you have the take this TB skin test and it tests positive, basically you are considered affected. And so as a result, they will test positive and have to go through this drug regimen if they wish to work in the United States. So the question, once again, is what is your opinion of the different countries' different opinions, and perhaps what kind of barriers that's presented?
Yeah, so I mean, up until recently in the UK, we've routinely used BCG in adolescents because the British MRC study was conducted in adolescents and showed that BCG works. Rates of TB are relatively low in the UK, and in fact we've now switched to a program where we vaccinate infants, at-risk infants. But there isn't a universal BCG vaccination program in the UK anymore. In the US, the parallel trial to the British MRC study showed that BCG didn't work. So that was a study done in Chicago. In fact, I think it's on my, if you go to my cartoon, let me just find, yeah, okay, so Illinois children, there you go. And Georgian school children. So you can see that, you know, in the US, they conducted a study a similar study to the UK in adolescents and showed no effect of BCG. And that's the reason why BCG has never been licensed for routine use in the US. There are other problems with BCG, which is, of course, as you've just indicated, it interferes with the skin test. So you can develop a positive skin test if you're infected with TB, but also if you've been vaccinated with BCG, or if you've been exposed to environmental mycobacteria. There is a way around that now, which is that we have these new diagnostic tests, or IGRAs, for T-spot TB and quantiferon gold, which allow us to distinguish between BCG vaccination and latent TB. So I think that is no longer a sufficient reason to withhold BCG. BCG is now is pretty much given throughout most of the world. I think the best evidence that BCG works is when it's given at birth, and therefore I think the best time to give it is at birth. And I think you know, it's justifiable to give that in areas of the world where TB is a sufficient problem that the cost of that program therefore merits it. I think these end up being cost-effectiveness analyses, and actually if the prevalence of TB is too low in any population, then that's why it isn't, BCG doesn't end up being given. But I think the reasons in the US are historical and have to do with the fact that that trial, which I've just indicated, didn't work in the US.
All right, thank you very much. The next question is, what is the primary source of funding for most of these TB trial programs? For example, is it the NIH, the National Institute of Health, private donations, perhaps organizations from Britain? The student doesn't exactly know the system as well for Great Britain and the UK, or is it something else?
Sure. So funding for these programs is always a challenge, and the issue is that, of course, there is a limited commercial market for TB vaccines. So the money is largely public. My program has been funded largely by the Wellcome Trust, a UK-based charity, but also from the European Commission. The Gates Foundation and ERAS, which is a Gates-funded PDP based in Rockville in Washington, are responsible for a lot of the funding for TB vaccine development globally as well. Those are the main funders, I think.
All right, thank you. The next question is more of a social question. What kind of cultural barriers and or blockades have researchers conducting trials out in the field actually in different countries encountered during their research? For example, taboos against receiving shots or something.
Sorry, you broke up a little bit in the last bit.
Oh, sorry. For example, if in different countries, perhaps researchers have, if the student asked if researchers have encountered any cultural barriers or blockades when they are actually conducting these clinical trials in different countries during the research. For example, the student lists if tribes have, certain tribes perhaps who were approached for vaccines have taboos against receiving shots or something else, or a religion issue or something.
Sure. So engaging the local community in these kind of trials is absolutely critical. And having collaborators and people that we work with who are from the local community and based on the ground is absolutely crucial. And the issues are different in different places. So in West Africa, we spend a lot of time getting community engagement. There are real issues in West Africa with blood volumes taken. So in our trials that we do, the volumes of blood we can take to test for immunogenicity are much smaller than the volumes of blood, for example, we take in South Africa. The trial site in Worcester is a trial site where SATB, the South African TB Vaccine Initiative, have been working for about 15 years. And they have an amazing amount of community involvement and engagement and spend a lot of time and effort talking to the local community, explaining what they're doing, holding open days, so that, they're really, there's a huge amount of effort put into being very open and very clear, because of course that impacts on enrollment. But I think the point is that the issues are different wherever you work, and you have to find out what the local issues are. They are different in every place.
Thank you very much. Wow, we actually have 3 more questions, in fact. So the first question here is, how much technology do these, for example, for each of these trials, there are tests to see whether there are basically people who are in the trial get TB, or if they have other forms of TB like latent or miliary TB. So the student is wondering, how much technology do these different trials have access to? For example, in some countries, perhaps there are access to chest X-ray machines that can take chest X-rays showing active TB in the upper apex of the lung lobes, or perhaps miliary TB with the diffuse black and white dot pattern, also shown using a contrast chest X-ray. And if there aren't access to this, I guess the definitive diagnosis of TB, which is, I believe, the chest X-ray, and what other measures have these researchers gone around to compromise with this?
Okay, so endpoint diagnosis in efficacy trials is crucial. You want to make sure you pick up every single case, but you also need to make sure that you are sure your case is a case. So the gold standard diagnosis for TB is culture positivity. So TB culture grown in the plate from sputum or any other bodily fluid. And that's why the primary endpoint in our efficacy trial was culture positive disease. Absolutely, it is necessary for any site where you're conducting an efficacy trial to have a certain level of infrastructure, which means radiology, where we can do chest X-rays. It means microbiology, where we can do cultures. All of those things are essential. Without those, we can't do an efficacy trial.
Thank you. The next question is, how have different countries' regulations for drug trials helped or hindered the progress of global studies? For example, different countries have different rigors of standards for how a drug will progress from phases 1 through 2 and 3 and even 4 of the trials.
Okay, so I think it's true to say that all of the first-in-class new TB vaccine trials have all been conducted in the country in which the vaccine was developed, which is either the UK or somewhere else in Europe or in the US. So in that sense, all those vaccines have then been through a European or a US regulator. And I think that's the easy way to do it, because then when those same vaccines with the same manufacturing process go to particularly Africa, where the regulatory capacity is much less, you know that it's okay because it's already been reviewed by a European or US regulator. So South Africa have the Medicines Control Council, which is the most developed, advanced, sophisticated regulatory agency within the whole of Africa, but it is grossly under capacity. I mean, it's very good. We've interacted a lot with them, but they don't have enough capacity for huge amounts of trials that are happening in Africa. I think there is a vaccine for hybrid 56 where the first trial was done in Africa, but very similar vaccines, so hybrid 1 and hybrid 4, had already been tested in Europe. So I think, that is a different. If it's not a first-in-class or first-in-man, then I think that's a slightly different scenario. But I know that this is something that the WHO feel very strongly about, about building up regulatory capacity within the developing world. And you know, the point is that we can't do efficacy trials for the TB vaccines in Europe or North America because there isn't enough TB. So, you know, if we are to get a vaccine that works, that ultimately might be used in Europe and North America, we have to test it in Africa because that's where, or India or China. And therefore, it is essential that we make sure that we don't compromise on regulatory standards in these trials.
Thank you very much. And the last question we have is a historical question. So this student asks, how effective does the current BCG vaccine that's used compare to the the original cowpox vaccine that Edward Jenner originally discovered from milkmaids, compare in regards to efficacy and safety?
Well, Vaccinia, which is the smallpox vaccine, which of course is the basis of MVA, modified Vaccinia ankara, was a very effective vaccine because it led to the eradication of smallpox. Now, part of that is because there was no latent phase for smallpox, whereas, you know, the latent phase for TB, I think, means that the prospect of eradication is something of a fantasy at the moment. Vaccinia was very effective. It wasn't without its side effects, particularly myocarditis, which is why people have led to developing attenuated strains of Vaccinia. But, you know, stockpiles of Vaccinia still exist throughout the world should there ever be the threat of the return of smallpox through bioterrorism.
All right, thank you very much, Dr. McShane, for answering all of those questions. Obviously, there was a lot of enthusiasm with your lecture. As we saw, there were in total 14 questions that the students asked.
Well, I'm delighted to hear it, and again, apologies if you couldn't hear what I said clearly enough.
Oh, no, it's fine. We totally understand. I mean, technical difficulties with the whole technology thing. Basically, the IHN team chose the best software that was available for these e-learning modules, so we're just hoping for the best results. This is our first inaugural class, so we hope that in the future we're going to do classes on AIDS and malaria, hopefully next summer, and we're hoping that we can see improvements in results and that with time technology will become better. Yes. So I guess in all, on behalf of the rest of the executive IHN team, I'd just like to thank you, and I'd like to thank you for telling us about all of this about the BCG vaccine and what's going on, as well as giving us insight into the latest research and what is, I mean, basically there's the general consensus that any textbook that we read is, by the time it's published, is at least 10 years behind in the research and everything. And here we're getting so much information about exactly what is going on currently. So we have information more about that 10-year gap. So we'd just like to thank you, and the students have really enjoyed your interactive lecture, and we thank you for helping us out.
No, my pleasure. Thank you. I fully support this kind of initiative, and I think it is definitely the way forward. It is definitely teaching for the 21st century. So thank you for asking me to be involved.
And thank you for basically educating the world's next generation of global health leaders.
Thank you very much. All right.
Please, Dr. McShane, please feel free to exit this meeting whenever you would like. Students, I have a few announcements to make before we break, and so thank you very much. Okay, students, please remember, thank you very much for getting up so early this morning. We totally understand. I mean, it's been crazy, so I hope you enjoyed the lecture moderated by Peter this morning, and that we have one more lecture. Once again, we totally understand. I mean, it's so hectic. We have 3 lectures today lined up back to back, and it's really crazy. The unfortunate thing was that basically we had to schedule everything, and this was the only time the presenters were able to fit the schedule. Like one of our presenters actually canceled his, well, I think, yeah, he canceled his lab meeting in order so that he could speak with us. And I mean, if anything, that's like, you know, closing your lab meeting in order to speak with another group. So thank you for cooperating, and we will take a break, and I believe at 12 p.m. CST, so I guess that's 10 a.m. PDT. We'll have our third and final lecture for today, and please feel free to grab some food right now or something. I know it's been really tiring, so yes, yeah, please feel free to go ahead and just exit whenever you want. Please, thank you very much. We apologize again for all of the, I guess, audio and visual difficulties. Like, we're basically trying our best to make sure everything goes smoothly. So please do let me know if you're having any troubles or issues seeing or hearing anything, and thank you for turning on your webcams when possible. So yeah, please feel free right now to exit whenever you like, and I'm going to close the meeting. And so the meeting will be recorded, and so if you would like to, you can watch it afterward. All right, guys, time for a break. See y'all. I will be moderating the next lecture.